So welcome everyone. It's my pleasure to
I've been invited already.
introduce Scott Tighe. I'm Stephanie Servetus. I'm an adjunct faculty here at the college, and Scott is a colleague and friend. So he hails from Burlington, Vermont. University of Vermont is where his home base is, but as you can see from his title, he spends a lot of time traveling around looking at interesting places. And the microbial communities that are there. So you didn't come to hear me speak. I'm going to turn it over to Scott and let him tell you about his microbial adventures.
Cool. Thanks, Stephanie. Before I get going, I just want to say thank you to Amy and Stephanie and Scott and everybody that invited me up. I got to tell you, what a nice place you have here. Don't take it for granted. It really is something special. Everybody seems to get along and everybody seems happy, although it's a little cold. But, you know, I'm from Vermont, so I'm used to it. But so anyways, I want to talk to you a little bit about Antarctica, the International Space Station, and everything in between. And it's basically doing microbial research on extreme environments. That's what I do. I work with extreme environments, and my appointment is in the cancer center, but everybody loves extreme environments and volcanoes. So these are some adventures, I guess you could say, that I've been partaking in for the past 10 years. So I'm going to frame this around the microbiome. It looks like you guys are totally involved with microbiome. I'm seeing these great posters by the undergraduates here, and they're doing microbiome profiling. I don't really need to go through this, but what a microbiome is here is we have more bacteria and fungi and viruses on our body, 10 times more, 15 times more than we do human cells. So everybody jokes and says, we're a minority on our own bodies here. So it's in your gut. Everybody knows about the gut. There's the American Gut Project, and there's all kinds of bacteria and fungi and yeast that live in your gut. And of course, they're in plant rhizospheres, they're on plant leaves, they're in soil, they're in the air. They actually make the world go around. So even worms that digest organics have a gut microbiome that's pretty sophisticated. So we have a chance of finding bacteria everywhere. So how do we study it? We culture it. We used to do this a lot back in the 80s and 70s. This is one argument I have why you don't work with food without gloves on. And there's a lot of culturing that we do, and we also do a lot of microscopy. This is aspergillus. These dark spores here are Stachybotrys. You see it. This grows on the backside of drywall, and the other green ones right here are cladosporium. So you can identify stuff by a microscope. But of course, what I'm going to talk about is DNA, and you can get DNA 20 million years old, and it's intact. You can sequence it. And there's some special procedures. Now, we've been talking about with ancient DNA with Josie. But this is what we're kind of going after. But we're looking at the microbiome. So what's there? This is a water sample that we had in Antarctica, but this is Cytophaga and Sphingomonas. And all of these bacteria, we want to kind of know who the players are in a microbial community. And that's what we're after. And when we know who's at, like if you look at this leg right here, we see the Coracocheles and the nostoc. Those are algae. So we know something already about this microbiome sample. And this is not high resolution. This is what we did back in 2015, and things have progressed pretty quickly over the years. So a little bit of review of DNA. A lot of you already know about DNA. There's 4 bases, the AGTC. So this is DNA strand. It's a double helix. And it's packed pretty heavily. You can't see this because we got Amber's little picture here. But these genomes, everything collectively put together is called a genome. So a human genome is 6.6 billion bases of DNA. We're diploid, meaning we have a set of chromosomes from our mother and a set from our father. Bacteria are haploid. They only have one set. They're a circular chromosome, and they're much smaller. So bacteria is about 4 million bases of DNA, and human is about 6.6 billion. But what we do in microbiome, and you've seen here at these posters, is we actually go in and we sequence the DNA. So what is DNA sequencing? So we have this stuff compacted in chromosomes, but we have AA, GCC, AA, TT. And we sequence these with automated DNA sequencers, and that's the sequence that we get out. We compare to databases that some of you might be familiar with, so we can actually go in and find out what an organism is based on its DNA sequence. So we do that with 2 types of sequencer. I'm going to keep it simple here. They're massively paralleled sequencers. So a short read sequencer, they only measure 300 bases at a time. So you can imagine 6.6 billion bases. If you divide that by 300, you have to do that by 300 million pieces. That's a short read sequencer, and you hear about that all the time. That's called Illumina. There's a singular and Element Bioscience, it's 300 bases. And then we have a long read sequencer and ultra long read sequencers that can actually sequence up to 2 million bases at once. Bacteria only 4 million bases, so it only takes 2 reads. That's what we have going on right here. We actually turned it on. And this is some leftover DNA that we had from the sourdough that Stephanie set up, and we turned it on. It's hooked up to a little power supply, so we can walk around with this, a DNA sequencer. So I bring this DNA sequencer with me. I just had it in Romania not too long ago, and I brought it down in the cave with me, and we did sequencing in a cave. So it's pretty portable. If you're so inclined, I wouldn't recommend it unless you're trying to impress your friends. So these are the sequencers. I think this is Stephanie. You have this. This is the Illumina short read sequencers. There are all types of sequencers out there, and they generate, this one, for example, generates 8 trillion bases of DNA overnight. So you get a tremendous amount of data. And this little guy will generate up to 150 billion bases overnight of ultra long read sequencing. But you can see here, we take it out in the field. This is Antarctica. I'm down there, and we're sequencing with it. So it's quite portable, easy to set up and run, but it suffers from accuracy problems. The previous one is high accuracy sequencing. This is 99.999% accurate, whereas these sequencers are about 99.3%. You think that's probably not a lot, right? 99.99 versus 99.3, but when you do that over 6 billion bases, that's a lot of errors. But we have bioinformatics, biomagicians, I guess you could say. They are computational magicians that are able to work with this data. So this is this device that's running right now. It has a piece of polyamide film with nanopores in it. And what it does is here's that the double helix that everybody knows about. And there's a little unraveling enzyme here called DNA helicase. It unravels it. It goes through this nanopore, and it goes through the nanopore here. It's generating signal, and it causes an impedance flux across the membrane. And that impedance flux is characteristic of the base pair A, G, T, or C that's going through. So that's how we sequence it. So there's a couple of ways to do it. One is we go after one gene. I highlighted this here in green. This is called 16S, but there's a couple of genes we can go. We just sequence this DNA only, and we take soil, we extract it all, and we say all the organisms that are in there, we only sequence that one little gene. It's enough of a sequence that will give us low resolution taxonomy, but you can do hundreds of samples very quickly. We, in the extreme microbiome, I'm going to talk about this in a minute, we do whole genome sequence. So we take the entire genome, we break it up into little pieces, and we sequence all of it. We get all the genes, we get all the organisms, we get the functional genomic clusters, which we can identify new lead compounds. Let's say for drugs, if you're looking for new antibiotics, this is the way you would do it from a DNA standpoint. Antibiotic resistance genes, people have heard of that. You can go and find out if there's antibiotic resistance genes in hospitals, let's say. And then dark matter. I'll talk a little bit about this. Go in and find new organisms that have never been discovered before. And everybody thought it's kind of like patents. 100 years ago, everybody's like, oh, every single thing that's ever been invented is already invented. They said in 1879. And now we know that's totally not true. And everybody back in the 80s said, oh, all the organisms that have ever been discovered in bacteria it's not true. We discover hundreds a week when we're doing this. So it's so sensitive that you can actually take 10 pets, 10 owners, and they pet their dogs and kiss their dogs. You can go and swab the owner and the dog, and you can sequence it, and you can pair them all up. The 99.8% chance that they can pair me up with my dog Daisy and you with your dog, Rio. So I just put these up because they're fun to have cute dogs during a presentation. This is my dog. She almost died. She had she had a major infection of her gut. She was very sick, and the vet didn't know what to do. So of course I got sequencers, and I said, okay, we're going to do her fecal microbiome. And we figured out that she had Clostridium perfringens, which causes gangrene and other types of diseases. And dogs, it makes them very, very sick and really gives them serious pancreatitis. So I was able to bring this to my vet. He looked at it and went, I don't know what this means. So we told him what antibiotic to put the dog on. But the power of microbiome analysis helped me cure my dog. But also fecal microbiome transplant, many of you have heard about, is if you have Crohn's disease, we can actually give you a colonoscopy and then replace your feces with approved clean feces. And actually, that's like a 90% cure rate on Crohn's disease. So that's a fecal microbiome transplant. We do a lot of those at our hospital. It's quite successful. Sounds a little creepy, but it actually has a really good success rate. So I just framed microbiome. I want to talk about the Extreme Microbiome Project. I started this in 2015. And what this is we go around the world and we look at these really exotic sites that do not have normal temperature, normal salinity, normal acidity. And we go and we try to find new organisms, new enzymes, and we use it to develop new techniques in the lab because they're the hardest possible organisms to deal with. So I just want to cover, I talked about dark matter. It's out there. This is Samantha. She's down in the Alvin sub. This is the bottom of a trench that is located in the Gulf of Mexico. And you could see here, it's covered up by amber up here, but over 12% of what we discovered here, even though we're able to profile all these archaea, 12% is dark matter. These are new organisms. So you just keep that in the back of your mind as I go through this, because we find new organisms all the time, and new organisms equal discovery and new product development. So in getting to these environments and actually sampling them correctly is not easy. It's not like just going to scoop up some dirt out in Sheridan. We need Alvin subs to go to the bottom of trenches, and we need helicopters to go to our sites. And then this is Yellowstone right here. Sampling water that's 100 degrees centigrade is not trivial when you think about it. We did this yesterday at Thermophilic, and we found out it isn't easy to go out and collect a sample when it's 100-degree water. You can't just reach into it. So we all the special tools for that make it quite fun. And all the special enzymes and technologies in labs so we can actually get through this is Yellowstone again. There's so much biofilm and slime with these types of environments, you can't even get to the bacteria. Here's just an image. Any professors in the audience that microbiology, you know that these capsules, this layer around the bacteria shield it so well. Trying to get to that bacteria is just one headache beyond getting through this biofilm that we have out here. So we have to invent all these products just to get to, and we've invented a lot of products in the extreme microbiome just so we could get DNA. But we also have to deal with something called the Nagoya Protocol and APHIS. I don't just go somewhere like Romania, which I've been. You can't just go and collect a sample and bring it back to the United States. It's illegal. You can get in big trouble. You have to have all the permits signed off by the U.S. Department of Agriculture before you can actually bring a sample back in or even purified DNA. Nagoya Protocol is something that I helped draft years ago. And what it says is if you go to Germany and you find a cave and you find some new dark matter, new microbes, and you find out those microbes can generate a new antibiotic, you can't bring it home and make $1 million on that without sharing the intellectual property rights with Germany. It's like fair trade coffee. It's very fair. So these are the sites that we have right now. And if you'll notice right here, this is Thermophilic, because we went there yesterday, and I'm adding it to it just to keep it fun because we're sequencing that, and we're going to see what lives in those hot springs. So these are some of the places we've been to. These are just great fun images. These are the acidic lakes out in Western Australia. These are a pH of 1.4. You can't get near this. It's so acidic. If you step in this water, it'll dissolve your shoes. It'll dissolve your pants, Josie. So don't go swimming in this water. But we went there looking for novel compounds. This is the Dankill Depression, Ethiopia toxic hot springs. This is boiling hot water. It's got sulfur. It's got acid. It's got calcium. Everything that every microbiologist hates for getting DNA from an organism is in this sample. That we study because we're trying to mimic what's happening on Europa. Because we're planning to go to Europa and collect samples, and that's a difficult sample. This is the Dead Sea. We've done this one, and we look for novel biosynthetic gene clusters or lead compounds that come from ultra saline environments. This is Vandy Joy, I showed you a minute ago. She goes down to the bottom of the Barrenness. These are called hyperbrine lakes. So what these are, if you're not familiar with them, this is the Orca Basin at the bottom of the Gulf of Mexico. It's about 10,000 feet deep. There are lakes at the bottom of the ocean. So it's hypersaline, so it's very thick salt. So underneath the surface, you have a volcano dome that's 100% salt, and it gets hot. And what it does is it melts, if you will, liquefies the salt. So you end up with a very, very highly salinity, and these are two phases. Like this is just regular saline ocean water, and this is hypersaline. And because it's biphasic, you could actually say, and I've seen the video, that this is the shore, and there's actually waves. So at this junction here, we have a ton of viruses, and the fish all have abscesses and melanomas and stuff. So we've been working on that, trying to figure out what is the trigger of these melanomas. I don't go down in that sub. I don't. I've been offered to, but I'm not that crazy. I think my wife would kill me. This is the Mobile cave in Romania. This is a sulfidic cave system off the Black Sea. We've gone to this 3 or 4 times, and what we're doing is finding a lot of new insects in this cave. But we actually have brought this device I took with me at the bottom of this cave a couple of years ago. We actually sequenced some of the water that's at the bottom of this cave. So it's kind of funny because I'm not a cave person, but this is this beautiful cave, at least from this picture. And the entrance is this little tiny concrete thing with a hole. And what we do is we lower you down 200 feet. And when you get to the bottom of this hole, there's a little tube, and that tube is this. And these are the new insects and water scorpions and stuff that are discovered. There's many of them. And this is a video. I'll see if I can get it to play. And just so you can see what actually happens down there there's all these tunnels, and it's limestone. And this is the cave expert that I was with, but kind of a neat system of caves. You can get lost down there very quickly. So these are sulfur caves. And if you know anything about sulfur, it's yellow. This is elemental sulfur. So the reason we're interested in this is because a lot of the extra exoplanets have a lot of sulfur on them. And I believe that there is sulfonated DNA, although I've been arguing with NASA people, I work with NASA, that if we want to go find life, I'll bet a coffee on it, that we have sulfonated DNA. So we are going and collecting the organisms that are living in elemental sulfur, and nobody's done it before. And what we did is we went and got that, we dissolved it in xylene, dehydrated in alcohol, then we resuspended it in saline, and we extracted the DNA. So there's organisms living in this stuff. This is the Blue Lagoon in Iceland. Has anybody here been to the Blue Lagoon in Iceland? Oh, okay. Well, this is nice warm water, 37 degrees, perfect for humans, not chlorinated. So you can imagine there's a lot of human stuff in there. So we went there and we collected some samples, and lo and behold, we found exactly what we thought would be there. Number one, Micrococcus luteus. That's one of the number 1 bacteria that are on your skin. And it's the first thing that pops up, 65%. And there was some other creepy viruses I don't want to tell you about, but it didn't stop us from going in and having a great time. It was just fun to collect a sample and say, yep, our analysis works. This is Stephen Green went over and collected these samples at the Doors to Hell gas crater. This is in Turkmenistan. We actually put a zip line across here, lowered him down. Notice how I said we lowered him down. I didn't go. He was with George Kahunas that you see on National Geographic TV all the time. They collected samples, and we actually profiled it. And actually, there are things down here, Nocardiopsis. All of these are thermophiles. Many of them are thermophiles. And some of them are actually bacteria that come from birds, which makes sense because birds go over it, they get warm, and they poop. So it was just kind of a nice story. We hadn't done much more than that. Was our first one. Here's something very interesting. This is Pitseti Lake. That's in Romania. And this is a friend of mine, Roxandra, and she's under there. And she went to the bottom of this lake, and it's all pink. And she said, what is this? So she contacted me, said, what is this? And I said, I don't know, get a sample, send it up. So we think we know what it is, but we actually have to get more of the sample and sequencing. But what a phenomenon. Nobody's seen this before. Nobody has ever seen this before. So I don't know if you would call it extreme, but it's extreme enough for me. We did the deep ocean of Greenland. This is us. We went out in a boat. There's a whole story behind that. But we actually were looking for diatoms, and we actually did discover a new species diatom, which is an algae, a glass-shelled algae. And here's a picture of the one. And this is a pipette tip right here that Dan, my tech, was going in, and he was under a microscope. He'd suck them out, and then we sequenced them one at a time on the sequencers. So the water here in Greenland, we're only probably about 1,000 feet offshore. It was 2,000 feet deep right here. So it makes you appreciate depth. I don't need to say much about Yellowstone. I think there might be a rival between Thermopolis and Yellowstone, but we did do that. It's very hot water, but It's not easy to collect that sample, but thermos, for those of you who have ever done polymerase chain reaction, the first organism in Octopus Springs is where they discovered thermos Taq polymerase. We went there, and the first organism we found was Thermus aquaticus. So, and we were doing this with NASA. We were testing a sampling device for NASA, and it worked out well. But you can't, if you've ever been to Yellowstone, it's hot. Like you can't be within 20 feet of that. It's like, how do you get a sample? So we had to take a piece of tubing with like a lead weight on it and DNA free, mind you, sterile DNA free, and throw it so it went down into the hot spring, and then turn on the pumps to sample it. Everything seems so easy on paper. So I had to add this slide in because we just did this yesterday, and this is Thermopolis, the hot springs, and we went out, we sampled all kinds of places there. Very interesting and quite different, actually. I'm intrigued by this because, not far up the road, you have oil horsehead pumps. I took note of that. So there could be some hydrocarbon bacteria in here. Biobacillus is there. So it's interesting. And we're getting cut off, but this is a team, and most of them are in the room right now. I just have to say you guys are great to work with, our young scientists of tomorrow. Fabulous. Tahiti, Morea. This is, some of you have probably been there. We did a top of a land to sea transect. We collected a sample every 1,000 feet. And it sounds like fun, but we did have to write a paper out of it. I wouldn't call it an extreme environment, but the prices of hotels are extreme. So we just got back from the Red Sea in the coral village. You might know the story about the whiting disease, the wasting disease of the coral villages that are happening. They put a KAUST, King Abdul University, is a very exotic university. They have a lot of money, and they're putting a tremendous amount of money into trying to figure out why the coral is dying, and they haven't had much luck. So we went over with a whole team, and we just sequence with some of our modern methods. And here's just stuff that I just got this off this morning, actually, as I added this, but it kind of interesting. This is pretty standard. Shineacoccus is there. There are viruses, but there's an awful lot of streptomyces, and some of them are uncharacterized. I didn't put the uncharacterized stuff on here, but we're trying to figure this out. And Raquel was just on Discovery last week. If you saw this thing, it was called Beneath the Surface. So she had this whole thing that we did when we were there. So all seems like fun, but there's a lot of work behind it. You can't see this very well, but this is the Lake Hillier, the pink bubblegum lakes in Western Australia, not far from Perth. It's 28% hypersaline, and we're looking for new compounds. And we didn't really find new compounds, but we did what we call biosynthetic gene cluster analysis. And if you look down here, you can't see there's something behind this, but you can see down here the cluster that we were looking for. We found a gene that codes for terpene, which is very popular in foodstuff. It gives you that, like the flavor that you get in like gin berries is terpenes. Hops have a lot of terpenes in them, and they also have preservative effects for foods. Underneath this that you can't see is bacitracin, which is an antibiotic that I'm sure many of you have heard about. We had an analog of bacitracin being produced by one of the bacteria in there. So you can go to these environments and find antibiotics. I just got a grant to go to Greenland snow to look for saccharophilic plastic degrading, plastic eating bacteria. Nobody had done this before. I said, geez, there's got to be bacteria that chew up plastics, because our number one problem with microplastics, they end up in Antarctica and they end up in Greenland, and they sit in these bays and they just float. So we went and spent about 3 years working on this, and we found 13 bacteria that could degrade microplastics. And we actually have that all, we're watching it degrade to plastic in the refrigerator right now. So we're really encouraged by that. We got 3 papers we published on that already. We're probably gonna, you can't see under here, but these genes, there's nylon dehydrogenase here, there's carbonases here. All of these genes are the genes that we're getting out of the organisms, the metagenomics, that are actually responsible, that are actively degrading the plastic. So we know it's actually happening, and we're watching it. We're culturing it. We're measuring the density of bacteria. So we have plastic degrading bacteria from Greenland. And this one's more for the students, because I want you to know that I still do gram stains. So these are the bacteria that we got out of Greenland. So Now the microbes that we're working on in zero gravity, we've been working on sampling all the spots in the International Space Station, in fact, and actually one of the bacteria that we found is resistant to a lot of antibiotics, and they named it after me. And it's resistant to like 5 antibiotics. I said, that's like a creepy thing that you're naming some bacteria that can kill people. So there's my name, Tighe. So it's named Tighe bacillus JPLEI. It was all executed by NASA JPL, so they called it JPLEI. So that was quite an honor for me. But I had a mission to the International Space Station. I built a DNA extraction device that's automated. It can work in zero gravity. This was our launch a couple of months ago. This is SpaceX 29, and we deployed microbial DNA, cells, yeast, and preservatives. And if anybody thinks that sending up stuff to the space station is easy, it's a nightmare. So this is what we built. These were all handmade. Everything from the screws, the plastics, the injectors, all of this I made with the help of 3D injection molds and 3D printers. This is the DNA extraction platform. It's a 3D printer that we ripped apart and we put magnets on it. So everything's moved by magnets. The DNA goes into, like, if this astronauts can take spit or blood and they can pipette it into this, and then the magnets will move it around through all of these chemical chambers, and it can be done upside down. We did parabolic flights with that. We rolled it on the ground while we were doing parabolic flights, and it worked just fine. So we know it works in zero gravity. And the big problem on the space station is they don't have any way to extract DNA. They have 5 of these on the International Space Station, and they use them. The problem is they can't extract DNA. So what the hell good is it? You might say. So this is our astronaut. This is Satoshi. He's actually the medical examiner on the space station. And we worked with him for 3.5 hours. And if you know anything about the space station, $180,000 an hour. So we had 3.5 hours for our research. And this is the device that we made. And you could see, and actually I got this as a video so you can actually see it. And it's a whole minute. So, So this is Satoshi. Now, remember, this is zero gravity. So there's a lot of problems with this. He's injecting the DNA into the valve. So we had to build these valves. And he's explaining to me why he's having problems, because it's easy to do it on Earth. And you can see there's no gravity. And everything like these tips, this tip box had to be engineered for the space station. It's got Velcro on it. When you open it up, it doesn't open and close. Tips don't go flying out. It all has to be certified, making sure that nothing can happen. So he's explained to us, we're doing space to ground. So we're on communications down at Huntsville and talking to him from mission control. So he has cameras, we got cameras, so we can see what's going on there. So it's quite an exercise to do it, and especially space to ground is kind of scary, actually, because everybody in the world's listening to you.
What was wrong? What was
So this little valve here has, when we designed it, has right about here has a cross piece of plastic and it's at an angle, so it can hold it in place, because you can't just have plastic holding the silicon in place, because the silicon are just pushed in. And he was going in at an angle and it was bumping into that plastic. And we were saying, Satoshi, straighten out the pipette. And that takes a while to say, straighten out the pipette. It takes a long time. You might have been, you get LOS, loss of signal. So you have to see all these things like, oh, satellites are out. And you just sit there and wait for 15 minutes until the satellites come back. And then you can talk again. And it's 15 minutes of wasted time at $180,000 an hour.
And what was the DNA that he's testing?
So what I did is I sent up microbial DNA from a cocktail of 10 microbes that are safe, because we can't send up pathogens. And then we sent up brewer's yeast as a control. So that was in one of these tubes down here that he was going to inject that. And in these are the DNA extraction chemistry. So
what did you find?
So right now, this is a technology demonstration to say that he can actually handle all this. So we did parabolic flight, which is, if you don't know what that is, you go up in a jet 757 and you do this 60 times. And on the descent, you're in zero gravity. But it's really hard. You only get 37 seconds. So you got to work really fast. So we passed that test. And now we're saying, can you pipette all of this stuff. And all of the stuff is new, like those valves are new, and there's a lot of new stuff. But it's actually a nightmare to work up there because everything's got to be Velcroed. Things float around. It's very hard to do.
One more time. Sorry. How did you make it so the pipette tips don't fly away?
So that's a good point. So when you open up the pipette tips, most aerosol tips, when you open up, will come out. If you take a pipette tip box and go like this, they just fall out. There's 96 of them in here. So what we did is we took a piece of plastic, we 3D printed it, and then we took a heater socket in it, made fingers on it. So when we put the tips in, you had to push them down with, it was like 41 micronewtons because we had to measure it. So he could go in and pull them out, but they didn't float out. And we patented it. I'm going to really get rich on that.
I don't know. I just feel a lot of
Yeah, that one you won't. So moving on to Antarctica, so you know there's some intrigue about Antarctica. This is me and Sarah down in the dry valleys of Antarctica. And what we're doing is, and this, talking to Josie without her, on her project over there, we're down looking for long-term survival gene. In Antarctica, they said the longest piece of DNA they've ever found up until 2015 was 800 base pairs of DNA. And we're like, phooey, longer than that, we can prove it. So we went down and we were looking for novel gene clusters and long-term survival genes because the organisms that we find down there are 30,000 years old and still alive. They're bacteria that are underground and they're just barely respiring. So we're in the dry valleys. So this is south of Christchurch, New Zealand. That's how you get there. It takes forever to get there. And right on the dry valleys right here, this is the driest desert on Earth. There's no snow. So this is how the dry valleys are. There's Farrar Glacier, Taylor Glacier. So these are the valleys, and there's lakes. So this is Lake Vanda. There's Taylor Lake. Right here, you can see it's red. This is Blood Falls. I'll show you a picture of that. This is Don Juan Pond up here. So there's a lot of glaciers, and every lake is very different. The salinity is different. The pH is different, and it's not easy to get to. So here's just some fantastic pictures of we're flying around in helicopters. This is the Farrar Glacier. This is the Taylor Lake Frixell down here. So this is us just flying along, looking for a place to stop and collect samples. And it was all predetermined. So this was our helicopter. I was down there for a month and a half. And when you fly around in Antarctica, these days are very long. The sun never goes down. So there's these little huts in certain places in the dry valleys. You could stop and get a cookie and a coffee, and then you keep going with the helicopter because you're renting the helicopter. And this is what it looks like down here. So this is the corner of Lake Frixell, and you can't see it, but right in here is the hut. And there's a glacier on every single one of these. But you can see it's gravel or stone, purified stone. This is Sarah getting into one of the National Science Foundation helicopters, and we chopped into the ice because at the bottom of this ice, there's these flakes of biofilm that have been there for 17,000 years, and that's what we wanted. But these are just some fantastic pictures. And you can see this is our gear. That's a big canister, so we are ways away. And the lake doesn't really look like a lake. But there's a lake there. Here's what we are after. These are ancient biofilms. So this is Lake Victoria, which is ancient. It's long gone now. But the microbes there are still alive, and they dry out, and we dig them up underground. And these microbes, if you can actually culture them. So this is going to Lake Bronsworth, and this is a different place where we're trying to get a different kind of bacteria. And we're trying to go to all these lakes, different lakes, and see if we can find different bacteria in these lakes. So the ground consistency is very different. The materials are different. I'll show you some other pictures in a minute. And there's a lot of cell phone service down there. I'm kidding. I was on a radium phone, but they give you a phone. And you can use it all day long. You can be on the phone for 4 hours if you want to in Antarctica. They don't care. But I called my wife and I said, hey, we're down in Antarctica. We're out in the middle of absolutely nowhere. But the funny thing about this is I'm standing there and my GPS, I look at a compass and it says south is that way. And if you look at your iPads, it says south is that way. And I'm like, what is that all about? Because we're right between true South Pole and magnetic South Pole. So you have to be so careful. You can get confused while you're down there. Just something that's silly. This is the Mount Boreas on the Olympic Range of Victoria Land. This is amazing because one is it's about 5,000 feet in elevation, and the structure is very different. The rocks are, they look like moon rocks, so they have holes all in them, like something's eaten through them because they look like they're limestone. But why we went up there is we once heard from somebody in Maine that there might be some moss that's very old buried up there. So we spent a day digging holes. This is David. He's digging a hole like a dog while we were up there, and he actually found something. This is David. So we're trying to be DNA free so we don't contaminate a sample. So the last thing we need to do is contaminate a sample and then have somebody that's going to review our paper like Stephanie or Amy and go, this is human DNA. You guys should have been wearing masks. So we're wearing masks and he's digging holes. And so you look at these rocks, look like they're moon rocks, or very, very cool actually. But lo and behold, after a day, we found this moss. It was down about that deep, and we found moss. And here it is. It doesn't look like much. When you find moss, it's pretty cool. So it's 14 million, not 60 million. I lied. So, but nonetheless, when we put it under the microscope, you can actually see the leaf structure. You can see pollen grains, I guess, and you can see the vascular system put on the SEM. It's almost intact. So this is amazing. So we actually took that and we actually sequenced it. It took us a long time to sequence, took a long time to get the DNA because the organism's DNA structure is very different. So most DNA from plants are like a 50% GCAT content, but this was 28% GCAT, which is really unheard of, which makes it very difficult to sequence in the lab. And it took us a few years to get a sequence on it. But this is what we were doing all over the place. We had these 3-foot deep holes, hundreds of them, looking for these little layers. And then we found these little layers. And these are biofilms. These are microbial biofilms that we had. So these is pictures under the SEM I took. These are diatoms. These are glass-shelled algae that are been buried for a long time. And then you can see all the microbes within that structure here. These are coccobacillus. But as we get a little closer, you'll notice the little dots, if you will, protrusions on these bacteria. These bacteria are very characteristic of Arthrobacter. Arthrobacter, you can culture them today from the soil, but they have that characteristic. So one of the things about they're gram-positive, that this just triggers you, and they have this like ribbing effect. And this is very old. So then I noticed under the SEM, we have these cube forms, and these are biogenic calcium crystals that the bacteria make. So these aren't from the ground. These are actually, if you zoom in further, you can actually see all the bacteria, and they make these crystals. And actually, there's a place up in Montana State that actually works on biogenic crystals. So this is very cool. And then I put it under EDS, X-ray diffraction, and lo and behold, we have the calcium peak here. So we published this paper. It's just so cool to see calcium biogenic crystals that were made, 14 million years ago by Arthrobacter. And we do sequencing down there. It's really a lot of fun because if you know, my hand was not warm, but we had the sequencer with us. So we try to do that when we go out to these places to keep it fun. Blood Falls. This is off of Lake Bonny, but this is about 50, 80 feet tall, this glacier. And this is iron oxides that come pouring out from an underground river. And we actually did metagenomics microbiome on that. And Gacilia came out. And this is a microbe that's integral to sulfate iron cycling. So it made sense that we were able to see that organism in that. We didn't have permission to pursue it past this. The NSF said that's all you can do is taxonomy, so we stopped there. But when you see something like this, that's the height of a 3-story building, you see this, it's called Blood Falls for a reason. You just want to get a sample and do something with it. So we went to Blood Falls. But all of these great organisms, Psychrobacter, cold-loving, more Psychrobacter, cold-loving, these are all of the things that we expected to see there. So getting back to the biofilm that I just told you, that we had something that looked like Arthrobacter that was producing calcium crystals or biogenic crystals. This wasn't taken too far from here. And when we went back and looked at that for the biofilm, the first thing that popped up was Arthrobacter. In fact, so many species of Arthrobacter, if you look here, and it was like, wow, I saw that on the electron microscope. It looked like Arthrobacter based on the morphology of it. It generated calcium crystals. And now we look at with the DNA evidence, it's definitely Arthrobacter that's coming from those biofilms. But it's not just frozen tundra down there. There are open pools in the cinder cones because there's a lot of volcanoes down there. And you'll be walking on the shorelines of McMurdo and you'll see this crevice, if it was cinder pool that actually has water in it. Because of the sulfates, and it wasn't cold. We were down there in their summer. And I said, I wonder what's in there. And lo and behold, we have oscillatory. We saw that today from Thermopolis. And then some Langbina here, some diatoms. And a lot of the pools are bright red. And it looks like somebody dumped bullet or painted. And it's this rotifer that has this red chloroplast in it, which I'd never seen a red chloroplast. I'm not a rotifer guy, but it's like, oh, that's cool. And there are millions of them in there. So if you don't know about rotifers, these spin and they eat algae. And right here, these are their jaws, if you will, but they're chlorophyll. So, and this is, he went with us on every single trip. We're in Antarctica, so we got this pet penguin. It's stuffed. Well, it's a toy. It's not a stuffed penguin. That would be totally bad. So I brought him back with me, and my dog played with him all the time. So I wonder, as we do all this stuff and the technology changes so fast, it's not just extreme environments, it's just extreme things you can do with DNA. And one of the things that's starting to intrigue me more and more is DNA origami. And it's not a joke. You can actually it's like, you could call it micro machining with DNA. You can actually form molecular structures with organized DNA targeting. So these structures are all built in DNA. And you would look at this thing, that looks like a virus. But it's not a virus. Somebody actually, I don't know who did this, but they actually made these structures. So you think about how you can manipulate your own molecules or build your own antibiotics or functional genomics to actually have something happen with things that you can make with DNA as the tool itself rather than being the thing that you're studying. So who knows what's going to happen in the next 5 years. So there's a lot of people who are part of this team. I just take credit for it all. So if you have any questions, email these people. I kid you not, I can answer most of them. But I got to say thank you to Stephanie and Amy and Scott and Michelle Turner who helped organize this. And it's been a great experience here in Sheridan. You guys have a terrific place. And I will tell you that take a minute to come up and look at the DNA sequencer as we sequence the DNA here. It's going through a refresh. So I will stop there, and I would be happy to take any questions.
Scott, what did you find in Vermopolis? Do you know yet?
Well, that's funny. We looked at it today under the microscope. So I could tell you just looking under the scope, I can see it looks like thiobacillus, which is a bacillus that likes sulfates, and it deposits sulfur granules in its cells. I saw oscillatoria, and then a lot of bacteria. So that's all we know, and that's just by microscopy. So we just got the samples, and it's a great tool that we brought. We can concentrate like 5 gallons of water in a few hours in a DNA-free format. So that's a problem that we've had up until about 2 years ago in microbiology. For us to concentrate water has been a real challenge because you have to take filter apparatuses and pour water and filter it through and take these membranes out and grind them up. And when we do this stuff, the last thing we want to do is introduce DNA. And we have a new device called an AnovaPrep that we brought out to try it out at that site. And it's changing our life, actually, when it comes to doing environmental genomics. So, but I know those two things are there, and maybe somebody else, I know Amy's been looking at it, and Steph's been looking at it, students been looking at it, but those are the two that I know there. We'll find out. We're going to sequence it.
For all these sites, do they have something in common, like are either for acidic sites or alkaline sites?
Yeah, so this project is really well known in the world. So it was a spinoff from the Earth microbiome, and Rob Knight did that at UCSD, and then I started Extreme Microbiome, and everybody talks about it. So people will call up and say, "Hey Scott, we got this site. It's a cave in Bulgaria that's got a whole bunch of bat poop." And they're like, "Yeah, it's not really extreme." but then there's people like we did the hummingbird caves in Costa Rica, and that was very interesting because they have a very different gut microbiome. I'm not just into volcanoes. The gut microbiome of a hummingbird is pretty intriguing, and the gut microbiome of a goat is pretty intriguing because these animals, like a goat, can eat anything. They can walk up and eat a car battery and he's fine. So, there's all these, that's an extreme site. A goat gut is an extreme site. So yeah, people just reach out and say, hey, is this an extreme site? And they want me to go down and sample. It's like, I don't have money to go sample bat poop caves, but so.
What's a typical grant that you get?
So a lot of these are covered by explorer clubs. So the Explorer Club is a very rich club where people like Jeff Bezos and those guys want to go along with us and say, oh, how do you sequence DNA in a cave? And they want to be part of it because they can spend money like it's nothing. Some of the larger grants like NASA and the Greenland grant were $850,000, not a whole lot, but it comes with a lot of work, right? Because we got to publish papers. And like the Greenland, we promised the Department of Defense that we would write 4 manuscripts, which is a lot to do in 1 year on top of the other manuscripts. So this year, I think we published 13 manuscripts and I wrote them all. And it's like, oh, it's fun to go to this. It looks cool until you got to sit down and write 13 manuscripts in 1 year going, what did I sign up for? I should have just stayed home. So.
You know if NASA has plan to include DNA sequencers in any of their spacecraft?
Yeah, so it gets brought up all the time. So I'm part of the planetary protection group. And so I'm very knitted in with NASA, and it's totally involved. It's happening. So if you're not familiar with Gateway, many of you are probably familiar with Gateway. This is the space station that will be located between Earth and the Moon. It's going to be a docking station for preparation. So that will have sequencers on it. And the spacecraft going to Mars will also have sequencers on it. And this was the call for bid of grants. We can't extract DNA in zero gravity. Somebody's got to do it. And that's what we answered to, but no question about it. On the fly DNA sequencing will be happening by NASA for sure. And they're doing it. They have the biomole group, and that's what they do is sequencing in zero gravity. But they don't know how to extract the DNA in zero gravity. It's going to happen.
So you had talked about a cave that was not previously open to air or exposed to air. I'm just trying to wrap my head around this. So it was some sort of talkative thing that air could not get in, and how would you go in and sample without introducing air into that cave situation?
Well, I think what I was mentioning in that regard was if I go back, and I'm not sure I remember it correct, which one I was talking about, but Mobile Cave, the one that I had the video of, that had never been open to air. 1989, a farmer and the guy that you see, the caver, that was their property in Mongolia. And he was putting a fence post in the ground and it went, it disappeared. So all of a sudden they dug this out and they found an entrance and they started going into the cave. And a lot of people were like, what are you doing going into that cave? Until like get professionals in there before you just hop in there. So they went in and ruined, if you will, the natural microbiome of that environment. The sulfidic cave, the one with sulfur in it, there's no oxygen in there. And it's biphasic. And if you take, let's say a cigarette and you just make smoke, it'll just sit at the middle and it'll just bounce, puff up and down. So what we do is we put on fully contained breathing gear and we go into it, but make sure you don't bend down below it because it's acidic. It's sulfuric acid vapors down here. But if you're up here, it's just carbon monoxide and hydrogen and stuff like that. So you can walk around, but there's no oxygen. So you have to have your mask on. That layer's right here. So you can imagine from here down, you got some funny sensations. Just saying. And I was like, this is really, they're telling me like, you're not going to like this, because down here is sulfuric acid. So, so it does burn you all around there, but you don't bend down to pick something up. It burns your face. So it's, you have these professional cavers. So we brought people in from Bulgaria, the cavers that you probably remember, the Thailand group. the kids that got stranded in Thailand and the cavers went in and rescued the Thai group, they're the ones that came with us. So we brought them up from Bulgaria. We just don't mess around with that kind of stuff. We bring in the professionals for it. Because you can get lost in a cave, and I've been lost in a cave before. And it's not, and my headlamp, everybody's headlamp died. It wasn't fun. So I'm like, not gonna mess with that one anymore. Scott.
Just a question about extremophiles. So you're going to look for in extreme environments, and often you'll find extremophiles, which I guess I've read about most of them. Hey, so are you discovering a lot of, probably, finding a lot of new.
Yeah. So the picture I showed you of this sub with Mandy Joy going down to the trench, those are all archaea. And the problem with archaea is the databases for archaea are not very huge. So for those of you that aren't familiar, there's bacteria, and there's a version of the bacteria before they were bacteria. They're called archaea, and they have different cell walls and slightly different metabolic patterns to it. And the archaea can survive extreme environments, super high temperature, super high pressure. The bottom of the Marianas Trench in the Pacific Ocean is 18,000 PSI. They can survive that. So it's all discovery actually at that point. But where do you start? We hire bioinformatics people to take the DNA and put it back together, and we try to build a synthetic genome from it, as you know, and that takes a lot of time. And I know that some of the students here are interested in that. So, but it's always a mixture of bacteria and archaea, but it depends on how like deep ocean water. When you go to Marianas Trench, it's like all archaea, except for stuff that sank down like whale poop or something.
How do you sample it?
You send this guy Josh that has the Challenger sub, and he goes out and samples it.
What's the vessel look like? Is it like
it's
a cup on a claw and he just like scoops it in?
It's, right. So the way they sample it is really difficult, and many of you probably know this. There's the hydraulic arms. There's 2 of them, and when you go to the Marianas Trench or even the Orca Basin, 13,000 feet is deep. I don't care where you're at. It's all such light sediment that you have to move ever so slow. So that last 10 feet, it takes them a long time to get to it. And then they sample with a hydraulic arm. In fact, Mandy brings styrofoam cups from her 3rd grade class. They tie them on the back of the Alvin sub. They go down 13,000 feet. They do their microbiology, collect samples with the hydraulic arm, and then they come back to the surface. And then the kids are there because, you know, it's their job to teach our young kids, and they get their cups back, and they're just crushed into oblivion, just like little pieces of plastic when they get them back. And I was like, not really. And then I went and saw one. It's like, wow, that is what I call high pressure. And then she's like, do you want to go down in the sub? I was like, yeah, no. I saw the abyss. There's no way I'm going to go down. And that guy with the joist, what was that, a Game Boy? Was that guy that was part of the Explorers Club that had collapsed on him last year? That was just total idiocy.
And you mentioned that you thought that you knew what the pink was in Romania. What do you think that?
Okay, so this I'm going to go out on a limb a little bit because I do need genetic information on it. It's actually a type of very rare freshwater jellyfish that produces a carotenoid, and it happens at very certain times of the year under certain conditions before turnover. But going into that lake, it's controlled by the Romanian government, and there's only a couple of people that have access to it, like Roxandra has it. So we're still working on that, and it's so neat. Like she sent me that, I was like, oh my God, that's so cool. I got to get that. So and they don't have the equipment in Romania to do this. That's not their thing. I've been over there like 10 times training them how to use this, and it's just they don't have the money for that over there. But they have cool sites.
What about the 12%? Because you talking about the 12% other.
The dark matter.
Yeah, are people?
Oh, absolutely. That's what, it's only as fast as bioinformatics people, computational people can assemble the genomes. But so Mandy, that was her project. We did it for her because we're the experts and we give it back to her. Now she has a graduate student that has a degree in computational biology. He's assembling the synthetic genome. And then it'll be their job to publish the papers in Science or whatever journal they want to do it in. And once they're done assembling the genome, they deposit them into NCBI, the National Database for Genomics, so we can go back in and find them. And I'm on the group that runs the databases. There's a big committee for that. So yeah, that's their job. That's what they get grants for.
And where's your next trip?
Where is the next trip? Wherever my wife tells me to go. So that's an extremophile Poland. I think we're going to do some stuff in central India. There's a desert there, but right now it's Wyoming right now.
Thermophilic.
Yeah. I don't know. It'll pop up. The email will come in, and I will say the Explorers Club wants us to go out to. Is anybody here familiar with Bikini Island? Bikini Islands in the South Pacific, or in the Pacific. It's where we did all our nuclear demonstrations, if we will. All the bombs were lit up. So you see all those pictures you see of like the mushroom clouds we did in 1942. That was all done on Bikini Island. So we will be at Bikini Island, but it's not until next year. It's next July we'll be at Bikini. We're getting the permits to go to Bikini Island. And it's in the Marshall Island chain, and they're very, at first I'm like, oh yeah, I'll just go to the Marshall Islands and get some samples and fill out Nagoya, but they got some chips on their shoulders out there. So we have to like be a little bit sidestep it, but that should be interesting. Now, this is an island chain. The Bikini Island itself has been blown to bits by nuclear warheads, both hydrogen and fission. So we're excited to go and check that. And obviously where we go, I will say, and this can happen to any of the students and researchers here that go to these extreme environments, for somebody to go to an extreme event, even going into a glacier and doing coring, you can pull out stuff that's 10,000 or 20,000 or a million years old. You can unleash things. Now, a lot of soil people don't respect that, but I gotta tell you, I worry about it because I bump into viruses that I can't figure out when I do this. So when people go out to these extreme environments, they just have to be a little careful, realizing that if you grow it up or you unleash something, you could have a problem. And the last thing you want to do is be responsible for wiping out the human race. It doesn't look good on your resume.
Right, yeah. How about an example of something that got monetized? So, something that we found.
That we found. I would say nothing yet. I mean, I got to tell you, as a scientist, I don't care about the intellectual property stuff that much. I mean, I have some IP, but I'm more into just discovery and let someone else take the IP. But I think the only thing we're working on is the microplastic degrading bacteria from Greenland. That is huge. And I think it would be smart for us to go to Antarctica, to the bays with all the plastics, and go down there and do exactly what we did and isolate the bacteria and fungi that are capable of doing it, and then bring those organisms up, get the genes out, splice them into better organisms. So see if we can actually start digesting some of the waste, because it's a problem. There's 500 metric tons of plastic in Antarctica. It's killing fish and seal. It's just ridiculous. So I think that's a big problem that needs, and I don't think it's going to be that hard. I mean, we've already isolated them. Just nobody wants to work with cold-loving bacteria that digest plastic. And I went and scanned the literature. There's one paper somebody worked on. And it's like, wow, this is.
So it digests it. What's a byproduct of the digestion?
So excellent question. So we don't do metabolomics. So that would be metabolomics, what happens after the DNA functions are occurring. But if we get another grant, we'll get somebody that does metabolomics. So this is genomics. You do metabolomics? See, I'll send it to Stephanie. I'm going to say carbon dioxide and water, but I'm sure that's not true. I'm not a protein or a metabolomics guy, so my expertise ends at RNA.
Any other questions for Scott? Feel free to hang around and ask some, but let's thank him again.